This week, the journal Science confirms something that sounds like it belongs in a thriller. Researchers used an AI model to design new viruses from scratch, and 16 of them actually worked. Headlines are calling it a landmark. Some are calling it a warning. Here's the catch: those 16 viruses only infect E. coli bacteria, not people. And no version of this AI was ever trained on anything that infects humans. So, how does AI design's functional viruses become both a medical breakthrough story and a biosecurity alarm at the same time? To untangle that, we need to know what these viruses actually are, why biosecurity experts and geneticists are reading the same results so differently, and what, if anything, actually changed this week that didn't already exist. The number worth sitting with is 302 to 16. Researchers generated hundreds of AI-designed genomes, and only 16 actually assembled into working viruses, roughly a 5% hit rate. That's not AI invents life on command. That's a model succeeding occasionally on the simplest possible target, an 11-gene virus, one of the smallest genomes in biology. Some of those 16 did replicate up to 65 times faster than the natural virus they were based on, which is genuinely striking. But faster at killing E. coli in a dish isn't the same claim as capable of designing a human pathogen. Here's the counter-argument, and it's a real one. Johns Hopkins biosecurity researchers warned in the same Science issue that the method itself, not just this specific virus, is the danger, writing that the ability to compose viral genomes with AI now exists, while the governance to safely steer it does not. That's a fair point about the technique, not the object. But test it against what a geneticist at Imperial College actually told The Guardian. These phages are, in his words, about the easiest genome anyone could make. And the real safeguard isn't stopping the AI, it's controlling who can order synthetic DNA in the first place, which quietly relocates the entire risk conversation. This was never really a story about a smarter AI. It's a story about a much older, much less discussed choke point: the handful of companies that turn digital genetic code into physical DNA. Zoom out, and the real story isn't this specific virus at all. It's a regulatory gap Congress has already flagged. A bipartisan bill introduced in February would make DNA synthesis screening mandatory nationwide. But analysts reviewing it noted a specific weakness. It screens by matching sequences to known threats, and AI-designed genomes can be functional yet novel enough to slip past exactly that kind of check. We don't know if that bill passes or whether function-based screening, checking what a sequence does, not just what it resembles, becomes real before the next capability leap. Watch three things: whether Congress moves this bill past committee, whether screening standards shift to function-based detection, and whether other labs publish similarly capable models with fewer safeguards than Evo's deliberate exclusion of human pathogens. So go back to that 302 to 16 number. It was never really about whether AI can build a dangerous virus today. It's about whether the guardrails keeping today's narrow success narrow will still hold the next time the success rate isn't 5%.